Morning, everyone. My name is Robin Bear with Whiteboard to Boardroom. We want to welcome you to our January webinar. I'm going to turn things over to James Baxendale, who will introduce our first presenter in technology. Jim?
Morning, everyone, and welcome to our UMKC Innovation Center Whiteboard to Boardroom webinar series. My name is Jim Baxendale. I work with the Whiteboard to Boardroom program. For some of you who may be new to our webinar series, the technologies presented by our institutional partners are available for licensing to form new startup companies or new product opportunities for established companies. In some cases, we'll also feature newly formed startups from our university and hospital partners. Those startups are oftentimes seeking CEOs, strategic partners, prototyping resources, board members, advisors, those types of things. Our presentations will run somewhere around 10 minutes or so in length, and we'll have some time at the end of the presentation for any questions, but of course you can use the chat feature if you like. Following the presentations, you can contact us directly to facilitate introductions, or maybe you'd like us to request further information from our presenters, so we're happy to do that. Presentations are recorded, and we'll send you a link to the presentations in a few days. And we actually encourage you to share this information with your colleagues if you think there's some value in doing so. So our first presenter today is Dr. John Rosen, is a pediatric gastroenterologist at Children's Mercy Hospital here in Kansas City, also director of telemedicine and outreach. He's a professor of pediatrics at the University of Missouri, Kansas City School of Medicine, and a clinical assistant professor of pediatrics at the University of Kansas School of Medicine. He will be presenting on his novel non-invasive method and device for detecting involuntary colon contractions. Dr. Rosen, if you'd like to start.
Great. Thank you, Robin and Jim. I appreciate the opportunity to talk about this today. This is a device and technology that we've been developing at Children's Mercy, which is a non-invasive device to monitor the bowels. And feel free to also contact me if you have any questions later. So what is the problem that we're trying to solve? Well, we make clinical decisions all the time, in the hospital, out of the hospital. And typically we like to rely on data. And in the cardiopulmonary world, we have lots of data to make our decisions, but in the GI world, we don't have as good of objective or continuous data. Often we rely on subjective data. How often did you pass gas? Or we listen with a stethoscope, which is classic but imperfect. And the problem with this is this leads to inefficient decisions. So we may keep patients in the hospital longer than we need to by not recognizing a change earlier. And we may make decisions that lead to complications by relying on the wrong type of information or inadequate information. And there is currently no method providing objective and continuous or long-term monitoring of colon activity for patients in or out of the hospital. Next slide, please. And so we came up with a non-invasive bowel monitoring system. And the non-invasive system uses skin electrodes, so stickers, as you see in the picture here, attached to wires and leads, and it measures electrical bioimpedance, so changes in an electrical field that is centered over the colon, as well as EMG or muscle activity. This is a continuous and objective measure of bowel activity. And the second picture we see is an example of the current technology that we're using that collects the data from the leads and sends it wirelessly to a computer. And you can see it is already very small in its current iteration. It potentially can incorporate into existing patient monitoring systems. So at the bedside in the hospital, there are patient monitors giving a constant stream of information that we use to make our decisions. There is no bowel indicator on those monitors currently, and potentially this technology could provide a signal to those monitors that would be an extra piece of information for nurses and doctors to make their decisions. It also could be configured as a wearable device for ambulatory monitoring, such as bowel training at home. It can be configured to send different signals. So depending on the target audience is. If it's patients or their parents or caregivers, it might be a simplified signal. The bowel is more active or less active. And in a hospital setting, it might be a continuous, more data-driven signal like we have for cardiopulmonary monitoring. In addition, this product can be used while non-sedentary, so you don't have to lay in bed. And some of the invasive monitors we currently have, which we'll see on a future slide, you have to lay still in bed to get that information. Next slide, please. So what is the market use? Well, we can monitor colon activity, and this could be applied to acute or chronic medical conditions. One of the chief cases is post-surgical ileus. So after somebody has surgery in a hospital, we expect in certain cases, like with bowel surgery or orthopedic surgery, for their colon to fall asleep. And it takes a certain amount of time for their colon to wake up, at which time it's safe to feed them. So they don't have vomiting, abdominal pain, surgical complications. Ileus can last differently in every patient based on their risks, based on the surgery they had, and we have no way to accurately identify when ileus is resolved or when it's prolonged to help us make decisions. Inpatient monitoring could be used for other acute and chronic conditions, such as inflammatory conditions or chronic constipation and impaction. And certainly it would be a way to gather more bowel information from patients with limited communication that can't tell us how their bowels feel, that can't tell us if they're passing gas or if they feel like they have an imminent bowel movement coming. In addition, there's an outpatient monitoring market, and this device can provide a signal that leads to behavior modification for toilet training, for instance. I'm a pediatric gastroenterologist, so I see this often in kids. It's one of the chief reasons for coming to a GI clinic. And constipation costs probably, the data says, an extra $4 billion in pediatric care annually in the US. And we see things like the Squatty Potty having $30 or $40 million in sale to assist with outpatient toilet training. Could also be used in pharmacodynamic studies to identify when a drug that affects the bowels is most active or when a new drug, whether it's affecting the bowel or not. And there are correlates in animal health, certainly in the corridor in which we live and work. There is a large animal health industry that's focused on post-surgical care, again, providing safe, efficient decisions for animal recovery. Next slide, please. There are no non-invasive colon activity monitoring devices in use. There's nothing available currently. There are invasive devices that we certainly use in the hospital, but they are very expensive. They require anesthesia. They require catheters put in the operating room. And again, patients can't be ambulatory with the existing devices. And there are no methods at home to do this either. There are two companies developing non-invasive devices that are in the research stage. They do not have a product on the market, and those are listed here. They measure different signals than the device that we've created. There will be questions of which signal is most accurate, which device is potentially the lowest cost, and certainly the technology used in these devices is all a little bit different. There is an opportunity that one or more of the technologies may benefit each other and may be used in combination, but it is a pre-market competition. Next slide. And the status of the intellectual property for this device, Children's Mercy filed a patent application first in August 2018, and national stage applications were filed in February. And what we see is one of the images for one of the use cases that's in our patent application, potentially on an outpatient setting or an ambulatory setting, sending a signal to a computing device so that somebody can make a clinical decision, whether it's the patient, caregiver, or medical provider. Next slide, please. So the stage in development, we have done preclinical studies to identify the most accurate way to collect this information, but we are continuing to study what is the best way to use this device and what are the attributes of the device that will be most helpful. We've identified signals in different positions, different movements, for instance, hip flexion or moving, walking, that might affect our signals. And we've learned a lot about how to take those ambulatory factors out so that we can use this as an ambulatory device. And we've also studied this after ingestion of a stimulant laxative medication in the setting of children who are already in the hospital, comparing the signals that we're collecting to the signals that currently exist with invasive devices. So there is no gold standard, but we are collecting physiologic information to compare to what other physiologic information we're even able to collect at this point. Next slide. And so next steps in development, we certainly are continuing to optimize our electrode placement, our data analysis methods. We have good methods now, but depending on the use case, those need to be continued to work on. And we're comparing manometry, or an indwelling catheter measuring colon pressure, to the electrical bioimpedance data from our device. And we are starting a study at KUMC in adults measuring baseline characteristics of what information does our device collect before and after an adult gives themselves a suppository and has a bowel movement. So correlating more to an outpatient setting, and we're collecting that baseline data in a pilot study at KU. And really for us, we're looking to secure a commercial partner, somebody with expertise that potentially we don't have. I'm a pediatric gastroenterologist working in an academic hospital and don't have the engineering background or the business development background. And certainly there are a lot of those resources available within the hospital, within our innovation center, but we would benefit from a commercial partner. And I'm happy to answer any questions about this.
Dr. Rosen, I've got a couple here. Can you talk a little more about how this would be used on an outpatient basis? Take through a scenario, I guess. And then also you mentioned animal use. How would you see that happening? So maybe a couple of questions there.
Yeah, those are great questions, and they are a little bit tied together. So the initial use case where this concept came from was a parent who brought their child into our hospital and had an invasive temporary test telling us about the child's bowel function. And she learned a lot from that test, but it's 2 days in the hospital and very expensive, and you can't do that at home. She said, why can't you tell me this information about when my child's bowels are ready to move when I'm at home so I can help direct him to the toilet instead of just asking him all the time? And so one of the outpatient use cases is in toilet training. So if you have a non-invasive device, like the picture in our patent application showed, kind of like underwear or pants, and it sends a signal to a parent or a caregiver, the child's bowels are active now. Now is the time to make behavioral change, to send them to the toilet. We do this all the time in terms of behavioral management in the clinic, but we don't have any objective data at home to help support that. There was a use case potentially for animal toilet training, but I think in reality, the bigger use case in animal health corridor is a post-surgical setting. And there are expensive surgeries that happen in animals, and they have the same condition, a post-operative ileus, that humans have. And so it could help make those surgeries more efficient and reduce complications as well.
In terms of the competition and the signals they're measuring, have you kind of looked at what that patent landscape looks like at this point to see if there's any, I guess we'll just call it overlap at this point?
Yeah, I think we've done that in several ways, and that's not my expertise, but Marcia Molina at our hospital has a lot of expertise in that field. Our patent counsel also helped us look at what's out there. And the PCT that we filed gave us feedback on the patent landscape, and all of their comments were grade A, meaning that they don't think there are specific items out there in the patent landscape that would interfere with the patentability of the technology that we're working on.
Okay. I believe at one time you were looking at a kind of a startup opportunity, or are you looking for direct licensing, or whichever one's most attractive, or what's that look like?
Yeah, I think whichever one can help us bring this product to market is our goal, to bring this product to something that can be used for patients. I think the goals would be a licensing agreement primarily, if that's possible. I think that the possibility of a startup is not ruled out in any way, but that would require partnership. That's not something that we currently have experience doing, and would require more expertise and assistance.
Okay. Are there any other questions out there? Looks like
I see some in the chat. Can I address those?
Yeah, sure, absolutely.
So there was a question, is this technology just for a healthcare environment, or could it be home use? And I certainly think home use is possible. We've focused on our initial use case in the healthcare environment because I think our ability to study it in that setting was easier to start with. But absolutely, once we have more information on the validity of the data that we're collecting and how it might apply to different populations, a home use case is necessary because there's a need and there's a market. The second question was, does identifying post-operation ileus potentially reduce readmittance? And yes, so how does somebody make money by identifying postoperative ileus? There are many different ways they could do that, but the financial advantage and the safety advantage is preventing complications, not sending a patient home too early, or in the hospital, not taking action like feeding too early. Because postoperative ileus costs a lot when it's not managed effectively, in the billions of dollar range in the US annually. And so if we can reduce hospital stays and reduce readmission, then there certainly is financial gain, and the value of the care is improved. So the patients will do better as well.
Okay. Are there any other questions for Dr. Rosen? Okay.
Thank you very much. And again, I'm happy to answer questions by email, or if somebody contacts through Whiteboard to Boardroom would be great.
Okay. Thank you, Dr. Rosen. Okay. Next up, we have two professors from the University of Kansas. Dr. Jack Tremble is a professor of practice in biotechnology and assistant director of biotechnology at the University of Kansas, KU Edwards campus. Dr. Randy Logan, professor of practice, academic program associate and director of the KU biotechnology degree program. So they'll be discussing their new diagnostic test for infectious disease. So if you gentlemen would go ahead, that'd be great.
Great, thank you very much. So before I begin, I wanted to thank Jim and Robin for inviting us to present here today. I also wanted to thank Ashwini Betha at the Center for Technology Commercialization here at KU for their support through the project. So as Jim was saying, although I'll be the one speaking, Randy and I have recently established a startup, Affinity Technologies, which is a collaboration between the two of us. We also collaborate, we make up the whole biotechnology department at University of Kansas, located on the Edwards campus. So to begin, we have a problem. We have a problem in how we as a society detect and respond to emerging infectious disease threats. I think that this has become very obvious over the past year, what sort of problem we actually have. We were actually caught pretty flat-footed by COVID-19. Even when we identified the organism causing the disease and sequenced its genome, we were unable to engage in rapid testing in order to do effective monitoring and contact tracing. And while we worked on developing a test of this sort, the world experienced uncontrolled spread of the disease. And we can see this here on this graph, where what we're looking at are new cases each day worldwide. So early on in the disease, we were having just a handful of cases per day. And as of last month, we were seeing 750,000 new cases a day worldwide. Next slide, please. So the trouble we have is that there's a 7-month development time in the best rapid response, easy to use test for COVID-19. So I'm using COVID-19 here as an example, but we're really talking about any emerging infectious disease now. The genome of SARS-CoV-2 was actually sequenced on January 10th of 2020. And it wasn't until the 20th, it wasn't until January 20th that we actually saw the first case here in the United States. Over this 7-month development time, we went from 1 and 2 cases a day early on in infection to up to 40,000 new cases a day uh, by August 20th. And what happened on August 20th was there was an antibody test introduced by Abbott Labs, which is really simple to use. It's very much like a pregnancy test that you would use with sputum, and basically a line shows up to show you whether you have antigens associated with SARS-CoV-2 or not. So that's a great test. It's rapid, like a pregnancy test. It's simple to use. You can use it at home or in a clinic. And it's relatively cheap. Next slide, please. What we've been working on is a test that would come out a lot more rapidly, but shared these characteristics of being easy to use, not require specialty equipment, and provide a rapid answer for your test right away. What we look at is what we've developed is a nucleotide-based test that would specifically target a novel infection. And we think that we could develop this within a month of genomic sequencing. So when I said before, the genome of SARS-CoV-2 was sequenced on January 10th, by February 20th, and the first case was in January 20th in the United States. By February 20th, 1 month later, we were only seeing about 20 cases total in the United States. Now, one of the weaknesses that we have is we don't really know how many cases we actually had, because we were doing a pretty poor job of testing early on, and that continues. But from the data that we have, only about 20 people had the disease in the United States, which is our focus here, 1 month after the genome was sequenced and the first cases started to show up. So next slide, please. So as I was saying, we propose a test that directly detects the genome of the virus using its genetic sequence as a target. In addition to a faster deployment, our nucleotide-based test would be easy to use, require very little training, very little equipment, and has a fast turnaround from test to result. What we compare this to are the 2 tests that are out. So I've already talked about the antibody test that Abbott Labs developed, which has a lot of these features, but wasn't introduced until August. The other test is an RT-PCR test, which has a lot of great benefits. It was easy and to bring to market very soon after we knew the genomic sequence of SARS-CoV-2. But the problem was when you got tested, that sample had to go to a laboratory where someone had to process that sample using expensive equipment that wasn't going to be available in every clinic. And the turnaround time was 2 to up to, I was hearing 7 days over the summer for test turnaround. So our test would be fast to develop, simple to use, adaptable. This is one thing I didn't say, adaptable to new viruses. So we think that 1 month time period would be anytime we get a new organism that we've sequenced the genome, we would be able to develop a test that would come to market that quickly. Yeah. Okay, next slide, please. And thank you. Like I was saying, COVID-19 is just an example. But what we did see was there was a very high demand for a test very early on. And the test that was available was unable to give us results very quickly. The other thing that we've done while we were developing this test was we've come across several other innovations that we've used along with our tests that we also plan to patent and explore a little bit. One of these is a reagent labeling test that I've sort of illustrated in a cartoon here, where we would be able to use affinity tags that we've developed for our infectious disease test to label reagents that are used in a number of molecular tests, including flow cytometry, Western blotting, and ELISAs. And these are the common tests that are used in any clinical reference laboratory anywhere in the world. The next slide, please. So as I was saying, that we recognize that there's competition. The RT-PCR testing comes to market very quickly, but it takes days to a week to return your results. And as we saw on one of my early slides, in the meantime, lots of people are getting sick as people don't know that they're supposed to be quarantining. Antigen tests that are the gold standard for viral testing, like the pregnancy test kind of test I was talking about, these are great, they're fast, cheap, reliable. However, they take several months to develop. So the test for COVID-19 took 7 months to develop. And we saw that, you know, over that 7 months, we were getting 40,000 new cases a day. So there's obviously a window here within those first 7 months where we need a rapid test to come to market. Next slide, please. So the status for intellectual property, we actually have one provisional patent that's been filed on the diagnostic test, and we're in preparation for 2 additional patents, one on nucleotide-based reporter adapters that I was describing, used in a lot of clinical reference laboratories. And the other is an amplification reagent for our assay that we think we can use for our assay in order to really improve the limit of detection, but is also, it relies on a technique that we think could be used on a number of different immunological tests. The next slide, please. So what stage of development are we in? We've done early proof of principle tests, and we've been very successful with those. We've looked at our limit of detection for our assay, and we're very pleased with what's gone on so far. It's been a very clean assay with essentially no false positives or false negatives. The only thing that we really need further development on is that amplification reagent that I was talking about. In order to improve our limit of detection, we need to improve on that so that we can actually detect the amount of virus that is present in biological samples, so the physiological amount required for making a test. But that's well underway. We're working on that presently. Next slide, please. Oh, okay, so this is our last slide. What we're actually looking for right now in terms of what we need for further development are, we're looking around for a business partner who can help us work through preclinical development, validation, and bringing the product to market. We also have a couple other goals here, clinical connection for final validation. We really need to get our hands on actual samples, both positive and negative, from patients so that we can validate that our test is actually working. And the other thing that we're working on, as I said, we founded the company earlier this year, and we're working together with Sharp Hub, which we've recently applied to, in order to apply for some early grants, an STTR grant, which is like an SBIR grant, in order to give us money to get through some of these early hurdles that we've been identifying. So again, I thank you for your attention, and I can take some questions if you have them.
Jim, you're on mute. Jim, you're muted.
Sorry about that. I was muted. Yeah, Jack, I had a couple questions here, and there's something in the chat room as well. I believe you said it's Infinity Technologies is the name of the company?
Affinity,
Okay.
AFFINITIE.
And is there, what's the regulatory pathway look like for this technology?
So that's a question that I've only recently started looking into. It's hard for us to really tell. We know the agency that we have to work through is the FDA. Unless we've also really thought a little bit about animal health, in which case we'd be going through the USDA. Currently, there are emergency use authorizations in place for COVID-19, which really speed up the time it takes to get through the regulatory hurdles. But that's something that I really need to look into a little more. My wife is actually a consultant who's sort of in a tangential area, and she's going to be asking some questions of her colleagues to sort of help out with that.
Yeah. Also, you mentioned adaptable to new viruses. So
Yes.
Could you give just a little more detail on that and the flexibility?
So our technology is based on looking at the sequence of the virus that we're targeting. So as I was saying, once we know the sequence of the organism, we can identify areas of this organism that we can target. And then the reagents that we use in our test are actually very simply made by existing technology. So we're applying a lot of existing technology to our question in order to answer get this test made. So I think that it's easy to pivot from one organism to the next. It takes attention, but honestly, when I've come up with new organisms that we intend to test here, for instance, we've made our initial test to the SARS-CoV-2, but we plan on doing in-house testing using a related coronavirus that is not as deadly or anything. And in order to change to use that virus instead of SARS-CoV-2, it took me really an hour to design, and then it takes less than a week to get the reagents to do this, at least at a research scale.
Okay. I think there are a couple questions in the chat there, Jack, if you can get those.
So the first question was, is this technology just for healthcare environment, or could it be used at home as well?
No, that's actually.
Oh, sorry, that was his question. Let me see, sorry.
From Jim.
Because I was actually thinking that does apply to ours, and I'll answer it anyway.
Okay.
We were imagining a healthcare environment, maybe a clinic that could do this, but it's not unreasonable to think that this could be done at home, with something that wouldn't be more complex than really like a home pregnancy test. So which is the first one for me? Did I miss?
Was from Jim with Core Catalytics, yeah, and he's wanting to know about the accuracy results and the average turnaround time for test results.
Okay, so in terms of accuracy, what we've actually looked at here is looking at the target sequence. We have a synthetic virus that we've been using in the lab as our target, and we've been using a scrambled sequence as a negative control, and we've seen no background whatsoever from the negative control, meaning that negatives look negative. And what is the average turnaround time from test to results? So, you could do this In the laboratory here, it's taken a couple hours, but we imagine condensing this down to around, 15 minutes to half an hour tops.
The next one is from Ron, and he is asking, how long do you think it will take to commercialize, and what do you think the cost of the test will be?
Yeah, it might be too early for those questions. I'm not really sure how long it will take to commercialize. I'm a little too new at the game to be able to answer, to make a good guess at that. I'm sorry about that. What we think of the cost of the test, however, we think that it'll be relatively low priced. And I'm just going to give you a shot in the dark. I think that we could do it for probably $30 a test or so, once we're actually up and running. But again, that's just a guess for that. I haven't really calculated how that would work out in the real world. I've only calculated how it works out here in the lab.
Okay, are any other questions at this point?
Hey, Jim, this is Aswini. Have a couple of comments.
Yeah.
So Dr. Trammell and Dr. Logan have been working with our office for patent protection and commercialization of this invention. I just want to let everybody know that we are very flexible to work with anybody interested in collaborating with Dr. Trammell and Dr. Logan for commercialization of this invention.
Mhm.
As the inventors just mentioned, a company name has been registered, and inventors are very enthusiastic to license the technology and commercialize take the product to the market. So anything we can help to facilitate and anybody interested in collaborating either for a research partnership or regulatory advice, we are open for discussions. Just wanted to bring that to everybody's attention.
Okay, great. Thank you, Ashwini.
Yeah, so thank you all for your attention. As John was saying before me here, you can please contact me through Whiteboard to Boardroom. I'd be really happy to entertain any questions that you have that you want to take offline.
Well, very good. Well, thanks, Jack. Appreciate that, and Randy as well. So as we mentioned, any questions you may have, you can forward them on to us. The recordings will get to you in a few days. And if you have colleagues, others out there in the communities that you think may have an interest in what was presented today, feel free to share those. So thanks again for attending, and have a great day. Thank you.
Thank you.
Thank you.